Hello, everyone. Thank you for joining us for today's webinar. My name is Isha, and I'm the product manager with OptraSCAN. I'll also be your host and one of the speakers for the webinar today. Today, we're presenting OptraSCAN's PD-L1 Workbench using AI toolsets, and it'll be presented by Dr. Clive Taylor and myself. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into the question box in your webinar window. I'll bring them up at the end of the presentation, and we will also have time for questions at the end. After the webinar is over, in an hour or two, you will get an email to download the full webinar. Now, without further ado, let me begin the presentation. All right. So before we dive into OptraSCAN's PD-L1 solutions, let me familiarize you all with what OptraSCAN is all about. We're an end-to-end digital pathology solution provider. We're focused on delivering fully integrated, cloud-based, affordable desktop scanning systems. And along with digitizing the glass slides, we also specialize in viewing, analyzing, managing, and reporting solutions as well. In today's digital pathology space, we're known for our high image quality and affordability, and we take pride in achieving this level of finesse without compromising on the performance and quality. Now, coming back to PDL1, our PDL1 solutions, rather the automated image analysis approach, has been developed to read and interpret PDL1 expression on tumor and immune cells in a given tissue section. In today's webinar, the attendees will get an overview of the complete PDL1 solution to digitize, index, quantify, and correlate. The aim of OptraSCAN's PDL1 companion research toolset has always been to assist pathologists with an aided scoring tool to enhance their efficiency, accuracy, and reproducibility of pathologic analysis. Our tools are all developed to help pathologists deal with challenging situations and make their life easier. Moving forward, a quick insight to our end-to-end PDL1 solution. This is an intelligent digital pathology solution which comprises of digitization of glass slide, indexing, analyzing, and collaborating on a single platform. Yes, you heard that right. We have a solution which can not only digitize your slides, but analyze, interpret, and help the pathologist read them. Most of you might be familiar with our solutions, but those who aren't, we specialize in a whole range of scanners for different throughputs. And along with scanners, we offer software solutions for further processing and reading those high resolution scans. That being said, this solution aims to overcome challenges and limitations by performing automated digitization, analysis, and reporting that otherwise might be an extremely burdensome and time consuming process for the pathologist. So, here are some of the points of discussion for today's webinar. Challenges of PD-L1 evaluation and scoring in immune effector cells. Dr. Taylor would be shedding some light on the common challenges being faced by pathologists in PD-L1 scoring. The next points include demonstration of the PD-L1 IHC scoring using the scanner and the AI-based toolset. OptraSCAN's patented fully automated solution for interpretation of PD-L1 IHC expression. Score generation based on the number of PD-L1 positive cells concerning total tumor and immune cells. And lastly, the computer-aided region detection system for an automated detection and evaluation of tumor nest and tumor infiltrating immune cells. So here we have the presenters for today. First, it's going to be Dr. Clive Taylor. He's the consulting chief medical officer at OptraSCAN, and Dr. Taylor has been with us for a very long time. And today it's my honor to be presenting with him. So Dr. Taylor is a fellow of the Royal College of Pathologists, a diplomat of the American Board of Pathology, a member of the Royal College of Physicians, and the Royal Society of Medicine. He was also elected president of the US Association of Pathology Chairs from 1996 to 1999. He's been the president of the US Biological Stain Commission from 1994 to 1998. And he's also chaired an FDA advisory panel for diagnostic devices and radiology until 2007. So thank you so much, Dr. Taylor, for being with us today. And the second presenter, that's me. I'm the product manager at OptraSCAN, and I've been with OptraSCAN for almost 3.5 years now. I bring to the table extensive experience in product marketing, product introduction, and the overall management of a product's life cycle from conception to fruition. I'm strongly involved in assessing customer desires, requirements, and generating solutions that successfully meet those standards. My daily routine includes, working with majority of the stakeholders, cross-functional teams, including development, marketing, sales, legal, distributors, domain matter experts, and customers. So again, it is my pleasure to walk you through one of our innovations in the immuno-oncology space today. And that being said, I will now allow Dr. Taylor to take over the presentation and shed some light on some of the common challenges being faced by the pathologists for PD-L1 scoring. So over to you, Dr. Taylor.
Thank you, Isha. So my role today is really to give a little background to PD-L1. I'd like to begin by wishing you all well on this rather troubled planet today, wherever you happen to be. And we are basically, most of us in lockdown, and so the technology we're talking about today has suddenly found new and important application, as we are in fact forced in many cases to sit away from our usual hospital environment and still render diagnoses. PD-L1, of course, is one of the hot topics in pathology. It's almost impossible to be a cancer pathologist today and not to have learned something of PD-L1. In brief, PD-1, programmed Death-Ligand, is an immune depressing function that is upregulated in certain cancers. The cancers in which it's upregulated tend to be those which are hypermutated and therefore have neoantigens that would possibly render an immune response that would be active against the tumor. However, because they're overexpressing PD-L1, whatever immune response might be present is in fact suppressed. A number of years ago, investigators and big pharma recognized that it might be possible to block the PD-L1 blockade by using antibodies against PD-1 or against PD-L1, the ligand. Fortunately or unfortunately, a number of different antibodies were developed, monoclonal antibodies, and different companies pursued these antibodies in the US through the FDA for approval. The FDA mandated in these clinical trials that as the antibody went into clinical trial, it had to enter trial with a companion test. This recognized the fact that not all cancers overexpress PD-L1, only those that do overexpress it would be expected to respond to therapy, and therefore it was necessary to find out which cancers would respond to therapy or be likely to respond and which ones would not. The challenge for pathology was that several different antibodies were approved together with their companion diagnostic test, or sometimes a less accurate test called a complementary diagnostic test. A companion diagnostic mandates that you have to do the test in order to give the drug. A complementary diagnostic is advisory only. You do not have to do it, but it's recommended that you do, and it gives additional information. For us as pathologists, the challenge is that it means there are 5 or 6 or 7 different tests that we have to evaluate, and these tests are not interchangeable amongst the different drugs, the different vendors, and the different platforms. So we have to learn to read all of these. It's become an incredibly important cancer biomarker and is a huge challenge for pathology because we not only have to set up the tests, we have to learn to read them, and we have to do it efficiently. We can't afford to spend an hour reading each individual slide, but these slides are extremely complicated. So part of the goal of the Optra PD-L1 process is that it will help us speed up the process and get accurate and efficient results. So it's become a critical part of the management if any one of your clinicians in your institution wishes to use anti-PD-1 or anti-PD-L1 therapy with any of the various therapeutics that are on the market, you have to perform this test. And clearly, we need to be accurate and we need to be able to reproduce our results and do it efficiently. And right now, it's difficult to produce reproducibility, especially in the context of scoring immune cells. Some of the scoring systems require scoring expression on tumor cells only as a percentage. Others require also assessment of expression on associated immune cells, mainly macrophages, but also T cells. This is extremely complicated and very difficult to do. I just want to spend a moment or two looking at what that challenge is. So here are 4 representative PD-L1 immunohistochemical stains where PD-L1 expression appears brown with diaminobenzidine. And the question for you, the pathologist, for us, the pathologist, is what's the score? And what is the threshold for the particular test that we are being asked to perform? There are some tests that have a threshold of 1%, some 5, some 50. A 0 score means the patient does not get treatment. And depending on the test and the approved scoring method, a 1 or 5 or a 50% score would produce a recommendation for treatment. And just looking at these 4 fields, it's pretty clear that the bottom 2 are strongly positive and would be certainly above 5% and maybe above 50, but that would be arguable. The top 2 are more difficult. Are they 0? The top right could be 0. The positivity we see there might be just immune cells. So our challenge is, which of these are negative, which positive, which patient therefore would be treated? What about the immune cell contribution to the observed score? And can we all get the same answer? So let's just look at the challenge of one high power field represented here. So let's assume we're just doing tumor percentage. And let's assume for this particular test, the threshold is 5% for the patient to be treated or not. So one high power field, to get percentage positivity, our numerator, the number above the line, would be the number of positive cancer cells, and the denominator would be the total cancer cells, positive and negative. So what's the total number of cancer cells? That's going to be our denominator. And how many positive cancer cells are there? That's the numerator. Well, in a single high power field, we actually really cannot count, even in one high power field, how many cancer cells there are. It's not possible to count them So we estimate how many it is. One can simply do the math, pi r squared for a cancer cell. Let's assume a cancer cell is 20 microns in diameter. And then in a single high power field, if the cancer cells were packed shoulder to shoulder, exclusively cancer cells, there would be 625. So I've just arbitrarily said 600, and in this particular field, it's obviously not all cancer cells. There's some connective tissue, there's some empty space. So I've estimated half the cells here are cancer cells, and that gives us a number below the line of 300. It's a guess, it's an estimate. So now all we need to do is the number of positive cells for a 5% threshold. How many positive cells do we need to see? Well, if you think there are 15 positive cancer cells here, the patient gets $100,000 worth of treatment. If you think there are only 14, they get nothing. Anybody think you can actually really do that? I certainly cannot. I can come up with a good estimate. Can I reproduce my score second time around? I've done this lots of times, and it's difficult to reproduce scores, particularly close to the threshold. It's quite easy to say, well, this 80%, 90%, or even 0. But when we come close to the threshold where 14 versus 15 versus 16 really matters, it's extremely difficult. And even if we get the denominator wrong, and it's instead of 300, it's 330, then of course the numerator changes. And 15 would be treated, for example, if the denominator was 270 instead of 330, etc. So this is very difficult. And counting one field by itself would take a long time. And even when we've done that, we've only counted one high power field on a microscope slide that might have as many as 2 million cancer cells. So we can see why this is so difficult and where some sort of automated assistance would be of value. We have a second problem, which is more recently been recognized. So here's another field. This is an adenocarcinoma of the lung. It's been stained for PD-L1. We can see there are a lot of positive cells. So if we counted the positive cells, that would be our numerator. Except that in this instance, we would be wrong because 40% of the positive cells, in fact, are not cancer cells, they're macrophages. And here now on the right, we're staining for CD68. And so the cytoplasm of macrophages stains red. And you can see about 40% of these cells have red cytoplasm. And if we go back to the left-hand field, here's a little cluster of macrophages down here. But some of these are morphologically indistinguishable or almost indistinguishable from cancer cells. Compare this little cluster down here to this cluster up here. This cluster are CD68 negative. Those are cancer cells. This cluster are macrophages. And as pathologists, we know we can't always identify every single cell. So we do a population-based identification, and we're quite good at that. But if we're going to score accurately, we have to go beyond the population to identifying individual cells. So this is one problem with the scoring process. I'll show you one additional slide that emphasizes the problem again. Here we've done it slightly differently. This is a squamous carcinoma. We've got positive staining for PD-L1. I asked a number of colleagues to look at this, and I got various scores from 1% to 5%, all of which in fact are wrong. Because if we now stain the cancer cell nuclei red, and we can do this because it's a squamous carcinoma with P63, then the positive PD-L1 cells down here, none of them have a red nucleus. These in fact are all macrophages, and the score in this case is actually 0. So this difficulty means that reproducibility for all of us is extremely difficult, and it's very time consuming. And any assistance we could get from some sort of artificial intelligence, computer-based assistant analysis would be of value. So let's just touch briefly on the test considerations, and then Isha will take us back through how the algorithm works on the Optra system. Now, I must preface all of this by saying if we don't have a satisfactory PD-L1 stain, then of course anything we do subsequently is not likely to be accurate. So there's extreme requirement for technical proficiency in producing the PD-L1 stain. We have to have a tissue sample that has been processed and prepared and sectioned in a satisfactory manner. And there should be some quality control of that process. That's a topic for a whole additional seminar, and I really won't go into it today. And then item 2 is the actual assay we perform, the IHC stain, in parentheses, is really no longer a stain under these circumstances. It's actually an attempt to be an assay, because we're going to try to quantify it. A stain, we just stain it, and it might be intense or light, but we don't quantify it. An assay, we try to quantify it. So this is a stretching immunohistochemistry to a new level of performance. We have to validate it. If you're using an approved FDA test, which is approved for a particular drug, one drug only, on a particular platform, one platform only, with a particular scoring system, one scoring system only, then you have to adhere to every stage of that validation process. Change anything in any way, and you no longer have a validated test. It turns out that about half of the labs in Europe and many in the US, although the number in the US is not as clearly known, in fact, are not adhering tightly to an approved assay, or are not using an approved assay at all, but are using an in-house lab-developed test. If you're going to do that, then you have to validate that in-house test against an existing approved test. And then once you've done that, adhere to your new protocol. So then step number 3 is really what we're focusing on today, because once we've spent all the effort on the test and performing the test correctly, if we don't score it correctly and reproducibly and accurately, then essentially all that's gone before is in vain. I am firmly convinced as an individual, personally, that we cannot score PD-L1 assays reproducibly, consistently over a period of time without computer assistance. So I'm a very strong supporter of digital image analysis moving forward. And that's really what the topic will be moving on. The Optra assay gives the advantage of being able to do tumor percentage as well as immune cell percentage, or combining tumor and immune cells to get a composite score. And these different scoring systems are part of the different approved assays that are out there. With that, I'm going to pass you across to Isha, who will continue from here on, and I'd be pleased to deal with questions at the end if there are any. Thank you. Isha.
Thank you, Dr. Taylor. And now the topic that everyone's been waiting for, the PD-L1 analysis tool set, and how OptraSCAN has been working to alleviate some of the above challenges described by Dr. Taylor. Before I present the tool to you, let me walk you through some of the major events that led to the establishment of this tool. Let's now talk about a study that was conducted to validate and assess reproducibility of the automated machine-based image analysis for PD-L1 IHC. The purpose of this study was to validate reproducibility of the automated image analysis approach. We compared conventional pathology scores for both tumor and immune cell positivity separately using the 22C3 mouse monoclonal primary antibody. 230 cases of non-small cell lung cancer were examined. Additional training and evaluation set of slides, including customized index tissue microarrays with variable PD-L1 expression, were utilized for training and validation of algorithm thresholds. For the study, we identified 5 micrometer sections representing 230 cases of non-small cell lung cancer, which was stained with 22C3 antibody on the Darko Link 48 platform for PD-L1 expression. These sections were scanned using OptraSCAN's OS15 scanner at 20x magnification with a resolution of 0.5 microns per pixel. A subset of 100 images out of a cohort of 230 were rendered viewable to 3 board-certified pathologists active in routine sign-out of these cases using the Optra web-based image management system. We assessed interpretation first by pathologists and second by the PD-L1 image analysis scores. Correlation coefficients were measured to assess the variability between the pathologist and also between the pathologist and the algorithm. Concluding the study, the automated scoring methods showed great concordance with the pathologist average scores that were pretty much comparable to interpathologist scores. So here are some snippets from our patented tool for PD-L1 analysis. This represents the input IHC image that you see on the top here, along with the outputs at the bottom, which contain colored overlays and legends representing critical components for PD-L1 analysis. Do not worry, all this will very well be elaborated and explained as I walk you through the tool specifics. So the algorithm basically determines the PD-L1 expression on tumor and immune cells. It shows partial or complete membrane staining on tumor cells along with membrane and or cytoplasmic staining on immune cells in a single read. The algorithm works on a whole slide. The whole slide gets analyzed and produces results for the whole slide analyzed image. So here's a quick representation of what the algorithm is expected to do. On the left, we have an input image, which has been analyzed to show PD-L1 expression. The image on the right is the output, which is a colored representation of the membrane in yellow, the positive tumor cells in red, the negative tumor cells in blue, the positive immune cells in green, and the negative immune cells or the lymphocytes in pink. As I mentioned earlier, the algorithm works on a whole slide, and that will be demonstrated further in the upcoming slides. Okay, so here's the interesting part. Let's now begin with a quick activity while presenting some of the intuitive features of the automated PD-L1 analysis tool. For this activity, we've identified a few interesting cases out of the many which were used for the above study. So here's an example. What you see right now is a 20x scanned region of an IHC stained non-small cell lung carcinoma sample. Let's observe this region for a few seconds before we apply any sort of analysis. Looking at the region, it looks like there's probably plenty of membrane, which indicates tumor positivity, and some blue cells, which may or may not be tumor. Well, when I say may or may not, that means there's a confusion. And why not? This confusion is something that a pathologist often experiences while scoring a PD-L1 slide. For me, I might score this a region maybe 70% positive. Well, I'm not confident, but let's find out. All right, so this helps. I've activated my image analysis tool, and at the bottom, you see some checkboxes. These checkboxes help the user look at critical components, either one at a time, multiple at a time, or all at a time. To get started with, I've checked the box for the membrane. It's clear that all of the membrane has been identified and picked up by the algorithm in yellow. That means most of the cells within the membrane have some positivity. But is it all tumor? Let's find out. Interesting. Now, if you see to your bottom, we're looking at positive tumor cells. Everything in red has been identified as positive tumor. But, oh well, when I take a closer look, there's still few cells within the membrane which are not red. Are they not tumor? Then what are they? Well, now I start thinking the 70% positivity that I assumed at the start might be a little off. My assumption was completely based on the consideration that everything within the membrane would be red. Well, if that's not tumor, then what is it? Let's find out. This is the most interesting part. There is positivity. However, it's not coming from the tumor cells, but the immune cells. Take a few seconds, observe the image, and you'll see a strong differentiation. And believe it or not, this is one of the major challenges today. And as Dr. Taylor has also pointed out in the previous slides, this still remains a challenge. Differentiating between positive tumor cells and positive immune cells or the macrophages. Even today, when I work with Dr. Taylor on some case reporting, scoring, etc. for PD-L1, there are instances when we're like, this might be tumor. Oh no, must be immune, but I don't know. Today, there is a strong need and demand for something that could help mitigate this challenge. Something which might be really challenging to the naked eye could be made easier with the help of such tools. However, coming back to the activity, my aim is to determine a score for the tumor and immune cells. Let's start with a tumor first. All right, now we've got rid of the immune stuff for a bit, and let's just look at the tumor. Clearly, everything in red is positive tumor, whereas everything in blue is negative tumor. And like the challenge of differentiating positive immune from positive tumor, differentiating negativity is equally challenging. As we see here, most of the cells which are blue have been identified as negative tumor. However, there are a few which haven't. And now it might be easier to guess what they are. Yes, that's right, negative immune cells or the lymphocytes. Even with a naked eye, the large, irregularly shaped blue cells are easy to identify and score. But what about everything else in blue? It's still challenging. And we'll talk about it in detail in the next few slides. Coming back to tumor, for the tumor score, the algorithm would utilize all the red cells as the numerator and the sum of red and blue as the denominator. The fraction would then turn out to be my tumor score. What about the immune cells? Similarly, immune cell scoring. What we see here are the immune cells, the green ones being the positive ones or the macrophages, and the pink ones being negative. The algorithm makes use of the green cells in the numerator and the sum of the two in the denominator. Along with looking at the components individually, the software or the application also allows the user to look at all of them at one time. So that was our first example. And you see how easily and intuitively the tool is able to perform something that a human still struggles with. So here's another example. This is a second case which has relatively less brown, meaning lesser membrane, thereby indicating lower positivity. Let's check it out. According to the standard protocol, let's activate membrane and disable everything else. The algorithm does manage to identify the membrane really well. Next, we want to see how much tumor is present. I see most of the inner areas within the membrane have turned red, but again, there are a few which aren't. And by now, I'm sure all of us know what they are. That's right, the positive immune cells or the macrophages. To validate what I just said, the green spots in the membrane are indeed positive. Let's go ahead and score the tumor like we did earlier. So here's a tumor. We've gotten rid of the immune cells, and in order to score, all my algorithm requires is the count of red, which is positive tumor, divided by a sum of the red and blue, and there I have my tumor score. Similarly, we do it for immune cells, getting rid of the tumor stuff, only displaying immune. For my score, all I need is the green cell count, or the positive immune cell count, divided by the sum of green and pink. And then I have my immune score. And lastly, why miss out on the entire canvas? So we have a select all checkbox, which would allow the user to beautifully capture all the critical components required for reporting and presentation purposes. Our interface also offers features like exporting the scores, capturing snapshots of the overlays for future presentations. It is an extremely user-friendly and intuitive platform, allowing the user to perform multiple operations on a single screen. Now I would like to emphasize a little bit on the advantages of this tool. I'd like to state here that no matter how hard the naked eye tries to improve, some challenges can only be overcome by the aid of a machine. And again, this is companion diagnostics. We've developed this tool to aid pathologists to make their lives easier and help them alleviate some of the major challenges today. The beauty of the tool is that it's always learning, and it presents a beautiful interface for the pathologist to report an accurate score. The second advantage is also its whole slide scoring method. Many a times, pathologists tend to miss certain critical areas while scoring the slide manually, and this could have huge consequences. And in order to avoid that, the whole slide scoring method ensures that all of the slide has been looked at and analyzed to present accurate outputs. This tool is a great aid, and again, it is never developed to replace a pathologist. The tool can be deployed on the web or on a laptop or a computer. It can work with images at various magnifications and formats. And to complement this tool, we always have our OptraSCAN whole slide scanners to digitize the slides and produce high resolution images at a minuscule cost. And that being said, I would now conclude my part and hand it over to Dr. Taylor for some final thoughts. Over to you, Dr. Taylor.
So, Isha, thank you. I've been watching the little chat box and some questions that are coming up.
Okay.
I think we can obviously try to deal with the questions. One of them I noticed really related to how did we establish, or how did Optra establish the ground truth as to what the score is and what the cells are. And it's a great question, and it's a key question. And the instrument and the algorithm is actually calibrated against pathologists. And the paper that you showed using the Yale cases from David Rim really used 3 pathologists who were experienced, actually 4 pathologists who looked at the slides, but 3 who were very experienced in relationship to PD-L1 scoring, producing consensus scores. And then the algorithm on a training set was tuned to match those scores as closely as possible. Once the training set had been used to train or teach or calibrate the algorithm, then the algorithm was turned on to a test set of cases, again, that had been consensus scored, And that was where the algorithm was evaluated as to how accurate it was versus consensus diagnosis and versus individual pathologists. So we have to be upfront about it. It's calibrated to the pathologist. So that is good in a way because pathologists have proved astonishingly successful in visual interpretation. I mean, we've been doing it for 150 years since the microscope came into medicine. And it's still the way of diagnosing cancer, in spite of all the molecular testing and NGS and the rest of it. Nobody treats anything without a pathologist giving a tissue diagnosis. So it's amazingly accurate, but it's not so accurate once we have to count. And that's because of the complexity of identifying individual cells and retaining numbers. So it's calibrated against a consensus of pathologists. It means it can never be better than the pathologists. It can only approach how good they are as consensus. Ultimately, of course, the real test of deep learning image analysis, which is beginning and which this is part, this process is a first or second step along the pathway. Ultimately, the image analysis would be set directly against the clinical outcome, and the pathologist, in a sense, would then learn from the image analysis system. That's already happening. There are studies being performed that show that image analysis in morphologic identification of tumor cells actually beats pathologists once it's been trained and then retrained against an ultimate diagnosis. So this is an iterative process. It's an ongoing process, and I've found it incredibly educational, both in terms of how good or bad I am at identifying cells and in fine-tuning the algorithm as you move forward. So that is a sort of a long-winded answer to a very difficult question. Are there other chat questions that we should deal with, Isha?
Yes, Dr. Taylor. So there's another question which says, what do you think was the reason that the interpretation of all the PD-L1 tests have been designed to be so complicated?
Well, yeah, I think in part, it's the process by which the pharma companies took these tests to initially the FDA. But clearly, there are approval processes in other countries, and I'm not trying to demean or diminish the CE mark in Europe and the Canadian process. They're all very good. It's just that the FDA sort of turns out to be the elephant in the room, as it were. It's complicated because what happened when the IHC test for PD-L1 was developed is that we saw brown staining on the surface of cells. And so people looked at the HER2 test, which was for breast cancer, which, if you go back and think about it, was actually approved in 1998, 22 years ago. And it was approved on a 1+, 2+, 3+, looking at membrane or partial membrane staining on cancer cells. HER2 was a very simple test compared to PD-L1, because PD-L1 is complicated by immune cell staining. And we've seen the difficulty in distinguishing macrophages from cancer cells. As a population, we can do it, but when it comes to, is this cell a cancer cell or an immune cell, so does it go into the numerator or not, on a single cell basis is very difficult sometimes. So it became complicated as during clinical trials, when the drug was being developed and the test was being calibrated to try to match which patients responded and which ones didn't, the pharma companies, together with their pathology vendor collaborators, were trying to develop an assay to give the best predictability. And it turned out that you had to incorporate both tumor cells and immune cells in some way in that assay, and different companies did it in different ways. Unfortunately, not everybody did it one way. It would be great if we had one antibody that worked, one test that worked, and we all did it the same. But unfortunately, that's never going to happen. So that's why it's complicated. It's the market that drove this, not true science in a way.
Okay, thank you, Dr. Taylor, for that. The next question is regarding the study. So They're asking if the study has been published and please send the reference. So to answer that question, yes, the study has been published and we have a full-fledged publication that we'd be happy to share with you via email after the webinar. So we have one more question for you, Dr. Taylor.
Yes.
Even when a pathologist is experienced with PD-L1 testing, how to distinguish every tumor cell from immune cell on H&E?
Well, the answer is we really can't. It's in some ways very humbling to go back and look at morphology. And you know, I've been doing immunohistochemistry for many years. I mean, our lab in Oxford was the first to do it, and the first thing I learned from it was, in fact, how poor, now poor is not the right word, how imperfect my morphology was in terms of cell identification. Because, you know, I would be, say, initially we were looking for plasma cells with immunoglobulin, and I was identifying with great confidence plasma cells morphologically and ruling out other cells. And then we stained for immunoglobulin and found that our morphologic criteria weren't very good. So having a separate set of parameters that helps in cell identification is enormously useful. It improves morphologic acuity, and it gives a second independent, more objective assessment. So I don't see this, as Isha said, as replacing the pathologist. Where I have found it most useful is getting the algorithm performed on the IHC digital image before I ever look at the IHC stain. That way, the ones that are clearly positive, you know, greater than 50%, the algorithm never gets those wrong. The ones that are totally negative, it doesn't get those wrong either. And if we take those two sets of slides out of the slide tray that we're being asked to look at as pathologists on any individual day, it removes a number of cases from our workload. We can then go look quickly at the slide, say, oh yes, the image got this right, the algorithm got it right, I can sign it out. And we can then focus our time and effort on the cases where the score is closer to whatever the threshold is for the test you're doing, 1%, 5%, or whatever. And that's where we need to spend the time. The algorithm does well on those. Does it get it right every time? That's probably not, but does the pathologist get it right every time? The answer is no. But putting the two together produces a result that is better than either one alone. And that's been demonstrated in other areas of digital pathology. There's a study called the Chameleon study that Google has been running. You can Google it and look it up, where they show that in morphologic identification of metastases of breast cancer in lymph nodes, that you could train an algorithm by deep learning within a relatively limited slide set, a lot of images to look at, and it would then match and beat the pathologists, you know, getting 96 or 97% right versus pathologists at 94% right. But putting the two together, you got 99.5% accuracy. So that's where I see the potential for this being. It's not a replacement for a pathologist. It's just an intelligent microscope that can help us perform at a higher level.
Okay, great. Thank you so much, Dr. Taylor. We have 3 more questions. The first one is, if user is using FDA's Roche PD-L1, will OptraSCAN solution be accurate as it was with Darko?
The answer is you would need to validate on a number of cases in your own institution the performance of the algorithm, and this is where Optra has its team to work with you to produce the scoring system for whatever PD-1, PD-L1 antibodies being used clinically and whatever platform it's being performed on with whatever monoclonal antibody, because the scoring systems are different and you will have to take the calibration, but the actual criteria that underlie the calibration are already there. So it's a relatively simple step to revalidate. There are issues with regarding the intensity of the staining that you develop, and if if you don't have really good control over your immunohistochemical staining process, including the hematoxylin component, then the algorithm can misinterpret some of the cells. So again, this can be tuned, but it does mean that the pathologist and the lab must pay serious attention to the quality of the immunohistochemical stain. And I have to admit, over the 30 or 40 years we've been doing staining, we have not paid enough attention to consistency in producing a stain of regular intensity. And that's where you have to use enhanced control processes to make sure the stains are reproducible. Because if your stains vary from day to day, so will the performance of the algorithm. And so actually will your performance as a pathologist, although you may not know it.
That makes sense. Thank you, Dr. Taylor. The next question is, the slides you scanned for the system training/tuning was created from the same staining, same company. So the answer to that is yes. All the slides which were used for the study were stained using the 22C3 mouse monoclonal antibody on the DARCO-Link 48 platform. So yes, they were stained using the same assays and from the same company. The next question might be for you, Dr. Taylor. What are the different immune cells that are counted? They vary in size.
Yeah, well, they do. Your morphology is a wonderful thing, isn't it? Because people that don't do morphology, including most of our clinical colleagues, think that the answer's black and white. The answer's 221 versus 119. And it's nothing like that. And we all know that. It's a matter of experience and training and evaluating what we see. It's a big problem with the FDA and with other approval agencies too, when they look at the results and try to understand what the issues are for pathologists. And there's a lot of concern as, well, if the quality of the scan isn't good enough, the pathologist might get it wrong. I think that overlooks the fact that as pathologists, part of our job is to, when we look down a microscope at a glass slide, if it's not adequate in terms of quality or stain, then we don't interpret it. We get another cut or another section or another stain. So we're not stupid. And this is the same with looking at scanned images. The scan has to have good quality and we have to be able to evaluate it. So when we then look at the cells, the immune cells have a wide spectrum of appearances. And we all know that. And when we look at any tissue section, we never identify every cell in that tissue section. We can't. I mean, if you take an immune cell, a macrophage with a nucleus that's 12 to 15 microns in diameter, that assumes that in the section we've cut it through its equator. If we happen to slice across the North Pole, then we might only see a fragment of that nucleus, just the cap of the nucleus, which might only be 2 microns in diameter. So that cell would be excluded from the algorithm as being unidentifiable as a macrophage. And incidentally, as a pathologist, we wouldn't have counted it as a macrophage probably either. So the algorithm builds in recognition criteria, and it will ignore cells that it cannot place within those criteria, which is a bit like we do as pathologists. It's more rigid, however, than we are as pathologists. With deep learning, algorithms can be trained even to identify some of those differences that I've just been talking about. So yes, it counts lymphocytes and it counts macrophages, predominantly those that have a representative cut through the largest diameter of the cell. And those that it can't identify with confidence, it ignores and doesn't score, which is what we do as pathologists anyway.
Right. Okay. Thank you, Dr. Taylor. And I believe we are down to our last question for today, which is this tool also useful for other IHC markers? So yes, the tool is definitely useful for other IHC markers, and we have a complete IHC workbench, which includes nuclear markers like ER, PR, Ki67, membrane markers like HER2, PD-L1, and also cytoplasmic markers. So this tool is definitely useful for other biopsy markers. These algorithms could be plugged in to the software and could be used at any point of time. And again, this tool can be hosted, as I mentioned, on the cloud or locally on the laptop or computer, and could be used by the user at any point from any location. On the last slide, we've also included our contact details. So in case you'd like to know more about these algorithms or would like to request for a live demo, we'd be more than happy to accommodate that request for you. So let me actually switch my presentation to the contact details
Yeah,
slide.
Isha, one
Yeah.
comment there is that
Mhm.
the algorithms as a set of tools can be applied to many immunohistochemical evaluations, but an algorithm designed for
Mhm.
PD-L1 cannot be used for HER2. You
Sure.
have to use the algorithm for the application for which it was developed. And for example, even if you're looking at nuclear stains like Ki67 versus ER, they're not translatable.
Sure.
They have to be validated for the marker in question and ideally for the platform and the immunohistochemical stain in question.
Absolutely. Absolutely, Dr. Taylor. Um, so coming back, to the presentation. So this is the contact. This gives you some information on the contact details, and we'd be happy to receive inquiries about this tool or even about the other product offerings that we have. We're still doing demos virtually, so feel free to shoot your inquiries to us, and one of us will definitely get in touch with you to understand your requirements in detail and schedule a live demo for yourself. So any final thoughts from your end, Dr. Taylor, before we wrap up the presentation?
No, I think it's outlined what the algorithm was developed to do and how it was developed. And I'm sure there are additional questions. My advice would be that for those that are interested should pursue it and try it with the basic caveat that they will need to do some internal quality control and validation in-house to ensure that the algorithm is performing the way that they would hope and expect in their own institution. But that's what we have to do when we set up any immunohistochemical stain anyway.
Absolutely, absolutely. And lastly, before we wrap up, in addition to leaving your inquiries to the contact details mentioned on the slide, there are a lot of resources and literature available on our website as well. So feel free to browse through our website. You'll find a lot of information about not only the analysis tools, but also regarding the different scanners that we have for bright field scanning, fluorescence, frozen section, etc. So when you get a moment, do visit our website and do contact us if you have any questions or require further information about any of our products. That being said, I thank you all for joining us for the webinar today. It was great walking you through, our different innovations in the immuno-oncology space, and hope to receive some more questions, some more inquiries, and hopefully we will get a chance to connect after today's webinar. So thank you so much, everybody.
Thank you, everyone. Stay well.